Now let's continue to understand the enzymes and the factors affecting its activity. So what are enzymes? Enzymes catalyze or speed up the chemical reactions without being consumed by the reaction. Therefore, it facilitates chemical reactions that otherwise would proceed too slowly to sustain life. So most enzymes are proteins, although some are made up of RNA, like the ribozyme. Ribozyme, it is a ribonucleic acid enzyme that catalyzes a chemical reaction. So the digestive enzyme amylase, for example, it breaks down the starch molecules into maltose, which are ultimately digested into glucose by other enzymes. So this is then absorbed by our body's blood circulation. The polypeptides and the proteins are broken down into peptides by the enzyme trypsin. Lipase, on the other hand, breaks down the fat molecule into glycerol and fatty acids. So now let's understand the structure of an enzyme. So take note that enzymes speed up the chemical reactions without being altered themselves. So in the picture, you can see an enzyme. So this blue colored molecule here, this is an enzyme. And this is called as a substrate. The substrate is the reactant of the enzymatic reaction, and together they form an enzyme substrate complex. So in most instances, so only one small part of the enzymes called as the active site associates directly with a substrate. So the active site is the part or is a part of an enzyme to which substrates bind to, and the substrate fits like a puzzle pieces into the active site. So once the reaction occurs, the enzyme releases the products. Note that the reaction does not consume or alter the enzyme. Instead, after a protein or after the protein releases the products, its active site is empty and ready to pick up more substrate. So, a metabolic pathway is a series of chemical reactions in a cell that build and break down molecules for cellular processes. So, in a metabolic pathway, it begins with a particular reactant or a substrate. So, this substrate binds to an enzyme, undergoing the first reaction, releasing now your product B. The product B becomes the reactant or a substrate that will bind to an enzyme, and that will take now reaction number 2, releasing product C. So product C becomes the substrate, binding now to an enzyme number 3, undergoing the third reaction, and releasing the final product D. So this is what we call as a metabolic pathway, a series of linked chemical reactions. So you already know that substrate binds to the active site of an enzyme, forming now the enzyme substrate complex. So two models have been proposed to explain how an enzyme binds to its substrate. So first, we have the lock and key model. So the lock and key model assumes that the active site of this enzyme and the substrate are equal shaped. So it supposes that the substrate fits perfectly into the active site of the enzyme. Second is the induced fat model. So when the active site of this enzyme comes into contact with a substrate, the enzyme will attempt to mold itself or induce now what we call as a conformational change around the substrate so that it can form an enzyme substrate complex. So enzyme speeds reactions by lowering the activation of energy or energy of activation. So what is this? It is the amount of energy required to start a reaction. So even reactions that ultimately release energy requires an initial kick to get started. So the enzyme brings substrate into contact with one another so that less energy is required for the reaction to proceed, just as it is easier to climb a small hill than a tall mountain. So reactions occur more rapidly if the activation of energy is low. Enzyme catalyzed reactions therefore occur much faster, millions to billions of times faster than they do in the absence of an enzyme. So now let's learn what are the factors affecting enzymatic reaction. Because enzymes are also very sensitive to conditions in the cell. So first we have the substrate concentration. So enzymatic activity increases as the substrate concentration also increases, just like here in this graph. It shows here that there is an increase of the enzymatic activity as the substrate concentration also increases. So if the pH is too high or too low, an enzyme can become denatured, or it will change now the shape, or it will alter now the shape of an enzyme and stop working. So therefore, this optimal pH varies from enzyme to enzyme. It is where this optimal pH, it is where the reaction rate is highest. So in this graph here, it shows that the stomach enzyme, which is the pepsin, works best or has the highest rate of reaction if it is in an acidic condition, while the trypsin, an intestinal enzyme, works best if the pH is in basic condition. So temperature is also important. Enzyme action generally speeds up as the temperature climbs because reactants have more kinetic energy at higher temperature. So in this graph here, human enzyme works fastest at the temperature of 37 degrees Celsius. However, if enzyme gets too hot or more than the optimum temperature, it rapidly denatures and can no longer function. So however, there are exceptions to these factors. So one of these is the environmental influence. So as you can see here, we have the Himalayan rabbits and the Siamese cat. So allele produce enzymes that allows pigment production only at the temperature of below 30 degrees Celsius, thus giving off their colored black extremities. Okay. And bacteria living in hot springs have enzymes that can withstand high temperatures. So, moreover, there is this what we call as enzyme inhibition, wherein it occurs when a molecule referred to as an inhibitor binds to an enzyme and therefore decreasing the enzymatic activity. So this enzyme inhibition has two general ways to prevent too much of a substance for accumulating. So in a non-competitive inhibition, the inhibitor binds to the enzyme at a location other than that of the active site. So this is called as the allosteric site. So thus changing now the shape of the enzyme and its function. So in return, it can no longer bind the substrate because it changes now its shape. Now alternatively, in competitive inhibition, the inhibitor and the substrate competes for the active site of an enzyme. It is competitive because the inhibitor or the product competes with the substrate to occupy the active site. So this graph shows that the rate of reaction increases as the substrate concentration increases with the normal enzyme in the process than when there is a competitive and a non-competitive inhibitors.